Good morning, students. In today's lecture, we are going to discuss reproduction in the reproduction in the bacteria. It takes place by the two methods. The first method it is the asexual reproduction, which is a very common method of reproduction in the bacteria, and the another method it is the sexual reproduction. Okay, the first of all going to discuss in this lecture about the asexual reproduction in the bacteria. Asexual reproduction in the bacteria it takes place by various means. Okay, here the different methods by which the bacteria multiplies. I have mentioned the different methods. The asexual reproduction, the first most common method of asexual reproduction in the bacteria is the binary fission. Okay, then second one is the budding, third one is the conidia, fourth one is the cyst, and the fifth one is the formation of endospores. Okay, these are the various of asexual reproduction in the bacteria. Then in the sexual reproduction, what happens? All you know that is male and the female gamete is formed. Then the fusion, that is the fertilization process of the male gamete and the female gamete get accomplished, and then there is a formation of the diploid cell takes place. Okay, that is the true sexual reproduction. Okay, but bacteria, when you observe that no such process takes place, that is in bacteria the formation of the gametes never takes place. Then the gametes are fused with each other. That means the typical sexual reproduction it is never reported in the bacteria, but the transfer of genetic material, which is the characteristic feature of the sexual reproduction, it is reported in the bacteria. That means the genetic material it is transferred from the one bacterial cell to the another bacterial cell without formation and without fusion of the gametes, and that's why the sexual reproduction in the bacteria it is referred as the genetic recombination. Simply we can say the transfer of genetic material from one bacterial cell to the another bacterial cells. In bacteria there are three methods by which the genetic material is transferred from one bacterial cell to the another bacterial cell, and these are: number 1, conjugation; number 2, transformation; and number 3 is the transduction. Okay, but in today's lecture we are concentrating on the asexual reproduction in the bacteria, and in the next lecture we are going to discuss about the sexual reproduction. Okay, today's topic of our discussion it is the asexual reproduction. Asexual reproduction in bacteria it takes place by various means like the binary fission, budding, conidia, then cyst formation, and the last one is the endospore formation. Okay, here of all, here you can see the asexual reproduction by the binary fission. Okay, the binary fission it is the simplest and the most common method of the asexual reproduction in the bacteria. Okay, it is the simplest and the most common method of reproduction in the bacteria. Under favorable conditions the bacterial divides into the two daughter cells. Okay, why it is called as the binary means the two. Under favorable conditions the bacterial cell divides into the two daughter cells by the formation of the transverse wall. Okay, bacterial cell, here you can see in this photograph, this is a one bacterial cell, and then there is a formation of a transverse wall between in the bacterial cell, and ultimately the two bacterial cells are formed at the end, and this process we called it as a binary fission. Okay, how exactly the binary fission takes place? Here the first of all the cell replicates its DNA. It is the first method, it is the first step in this binary fission. The genetic material or the DNA, that is it get replicated. Then the cytoplasmic membrane elongates, separating the DNA molecule. In this second step, cell elongation, cell elongation takes place, and that's why that is these genetic material, the genetic material DNA, that is it get separated from each other. Okay, it gets separated from each other. Then there is a formation of a median constriction at the center of the bacterial cell. Okay, just here you can see the formation of the median constriction at the center of the bacterial cell, and that median constriction then elongates. The median constriction then develops to the transverse wall at the center of the bacterial cell. Here you can see here, that is this median constriction, it is developed into the transverse wall, and later on these two bacterial cell, these are get separated from each other. Okay, and that is how the two daughter cells are developed from the one bacterial cell. Okay, the first step, it is the replication of the DNA. Second one, the elongation of the bacterial cell. Third one, formation of the constriction at the center of the bacterial cell. Then fourth one, the complete formation of the transverse wall between the bacterial cells takes place. And last one, the two daughter cells are separated from each other. Okay, here you can see the resulting bacterial cells, or the resulting daughter bacterial cells. These are the mirror images of the cell. These are mirror images of the cell. Okay, here the later on some scientists analyzed the cell wall components. They analyzed the cell wall components and observed that the chemical constituents of the original mother cell wall, it is equally divided cell wall of the two resulting daughter cells. Okay, some scientists, microbiologists analyzed the cell wall components and observed that the chemical constituents of the original mother cell wall, it get divided equally in the cell wall of the two resulting daughter cells. Okay, the process of the binary fission, it is a very rapid process. It is very rapid process because the bacterial cell, it may undergo the fission every 20 to 30 minutes. Okay, binary fission, it is a very rapid process because the every bacterial cell may undergo the binary fission every 20 to 30 minutes. Whenever the conditions are favorable, when the conditions are favorable, then in that case within the 6 hour, approximately 250,000 daughter cells may be formed from the single bacterial cell. Okay, if conditions are favorable, within 6 hours, about 250,000 daughter cells may be formed from the single bacterium. Okay, but that not happen because in the nature there are certain limiting factors. Limiting factors, for example, there is a nutrient availability. Okay, nutrient availability, and another limiting factor, it is that the accumulated waste products of the bacteria. As the large number of the bacteria are formed, the large amount of waste, it is accumulated into the mother cell, and that is how the nutrient availability and the accumulated waste products of the daughter cells restrict their rapid growth. Okay, that's why I have told you that is if the conditions are favorable, if the conditions are favorable, within 6 hours, about 250,000 daughter cells may develop from a single bacteria. But that does not happen because the nutrient availability and the accumulated waste products of the daughter cells are the important important limiting factors which are restricting their rapid growth. Okay, then that's why here we can say the growth and multiplication, it is very fast in the early stages. Okay, in the early stages because the large amount of nutrient is available and very few number of the daughter cells are formed. Okay, and that's why here the growth and the multiplication, it is very fast in the early stages. Okay, then later on it reaches the optimum level, and later their multiplication rate ultimately get declined, okay, due to the availability of the nutrients and due to the accumulated waste products. Okay, here I am repeating this one. The growth and the multiplication of the bacterial cell, it is very fast in the earlier stages, okay, because the large amount of nutrients are available, and as there are few number of the daughter cells, the accumulated products are lesser in amount. But later on, the multiplication rate reaches at the optimum, or it will reaches at the maximum level, and later on the rate of multiplication it get declined due to the availability of the nutrients and due to the accumulation of the waste products. Okay, then here the some of the students may get confused with the the binary fission. It may be just like the mitosis. Okay, that is in the mitosis also the one cell, it is giving rise to the two daughter cells. Okay, but binary fission, it is different from the mitotic division. Okay, why it is different from the mitotic division? Because in the mitotic division there is a involvement of the spindle formation. Okay, there is a involvement of the spindle formation when the nuclear material, it is equally distributed. Okay, in the mitosis there is a spindle formation, and also the another factor is the genetic material, it is equally distributed into the daughter cells in the mitosis. Okay, and opposite to the mitosis, in the binary fission the spindle formation never takes place. And another important thing, it is not known clearly, it is not known definitely whether the genetic material, it is equally distributed into the daughter cells. Okay, these are the two major differences between the mitotic division and the binary fission. Okay, in the mitotic division, formation of spindle takes place, and genetic material, it is equally distributed into the two daughter cells. But in the binary fission, the spindle formation never takes place, and it is not definitely known whether the genetic material, it is equally distributed, equally divided into the two daughter cells. Okay, that is all about the binary fission, which is the most common method of the reproduction in the bacteria. Okay, this is the binary fission. There in the another method, the budding. Okay, the second method of asexual reproduction in the bacteria, that is the budding. Here in the first photograph you can see the process of the budding. Okay, here you can see this is the bacterial cell. The first of all, here the nucleoid or the genetic material of the bacteria, it replicates. Okay, the genetic material first of all replicates, then there is a formation of a small outgrowth. Okay, there is a formation of the outgrowth, there is a formation of the outgrowth, and in this outgrowth the nucleoid and the cell organelles move into that small outgrowth, and now that small outgrowth, it is called as the bud. Okay, genetic material and the cell organelles, they get moved into that small outgrowth, and now that outgrowth, it is called as the bud. Later on, this bud, it get completely separated from the that bacterial cell, and one daughter cell is formed from the one bacterial cell. Okay, here you can see the first of all the nuclear material, it get replicated, then there is a formation of a small outgrowth on the bacterial cell. The genetic material and the cell organelles, it moves into the that outgrowth. That outgrowth is called as the bud, and later on the that young bud, it get separated from the mother cell, and from the mother cell only one bacterial cell takes place. Okay, thus how exactly the budding, it takes place in the bacteria. Okay, the examples of the bacteria in which the budding takes place, these are Hyphomicrobium vulgare. Okay, the example is the Hypomicrobium vulgare. That is a bacteria in which the budding takes place. And another one, it is the Rhodomicrobium vanilli. Okay, Rhodomicrobium vanillia. Rhodomicrobium vanillia. These are the two bacteria in which the budding, that is the type of asexual reproduction, it is reported. Okay, that is about the budding. Now the third one here, 1 minute please. Here, now the third one. I have mistakenly here written the binary fission, but actually it is the formation of the conidia. Okay, I will correct my mistake after completion of the lecture. Here you here read, please here read the formation of the conidia. Here you can see the conidia formation. It is a streptomyces. Okay, it is a type of the bacteria streptomyces in which the conidia formation it is reported. Okay, conidia formation it is reported in the streptomyces. Okay, generally the conidia formation it is reported in the filamentous type of the bacteria. Okay, we have already discussed the different shapes of the bacteria, and the conidia formation it mainly takes place in the filamentous type of the bacteria, for example streptomyces. Okay, it is the photograph of the streptomyces. Okay, it is a filamentous bacteria. It is a filamentous bacteria. Here you can see there are large number of the formation of the small spherical chain of conidia. Okay, these are small spherical structures which are formed in the chain like structures, and these are called as the conidia. Okay, the small spherical chain of conidia these are formed, and these are called as the conidia. Okay, and these conidia here you can see these are formed on the small filament, a small stalk like structure, and this stalk like structure it is called as the conidiophore. Okay, it is called as the conidiophore. Okay, small spherical structures these are formed on the chain, these are called as the conidia, and these are formed on the small stalk like structure, and this small stalk like structure it is called as the conidiophore. Okay, generally these conidia here you can see these conidia these are formed by the transverse wall. There is a formation of a transverse wall at the tip region of the filament. Okay, there is a formation of the transverse wall at the tip of the filament, and then that tip of the filament it get detached and develop into the new cell. Okay, these conidia these are separated from the filament, and later on each of the conidia will get developed into the new bacterial cell. Okay, that is all about the formation of the conidia. That is the third method of asexual reproduction in the bacteria. Okay, that is the formation of conidia. Now here in this photograph here you can see the cyst formation. The number 4, the type of asexual reproduction it is the formation of the cyst. The cyst formation here in this one you can see the formation of the cyst. Generally the cyst formation it is reported into the bacteria, for example azotobacters. Okay, azotobacter here also the photograph it is the azotobacter bacteria. Okay, it is the azotobacter bacteria. Okay, in this one here you can see what is meant by the cyst. Here a distinctive resting cell is formed. Here you can see the bacterial cell it is converted into the distinctive resting cell, which is formed by the deposition of the additional layer around the cell wall. Okay, here in this photograph you can clearly see there is a formation of the additional layer. There is a formation of additional layer which is enveloping the entire bacterial cell. Okay, cyst Simply we can say the cyst, these are the resting cells. Okay, the meaning of the cyst is the resting cell. In this photograph you can clearly see there is a formation of a additional layer. Now the bacterial cell, it is get converted into the cyst. Okay, here you can see the entire bacterial cell, entire content of the bacteria, it is get utilized for the formation of the cyst. Okay, it completely get utilized in the formation of the cyst. Okay, and this additional layer which is formed while during the cyst formation, it is resistant to desiccation, and it is not resistant to the heat or the higher temperature. Okay, that means generally the cyst formation takes place whenever the sufficient amount of moisture it is available for the bacterial cell, and the bacterial cell for their survival in the such conditions, there is a formation of the additional layer around the entire bacterial cell, and the bacteria it get converted into the cyst means that is the resting cell. Okay, that means it is resistant, the additional layer it is resistant to the desiccation. Okay, and it is not resistant to the heat or the higher temperature. Okay, that is about the cyst formation. That is how the cyst formation takes place in the bacteria. Okay, and here in this one, the last photograph, you can see the endospore formation. Endospore formation, that is the last method of the asexual reproduction in the bacteria. Endospore formation, here in the bacteria, endo means on the inner side. Okay, endospore. Okay, that's why here you can see that is these are the intracellular, these are the intracellular resting cells. Spore and the cyst, that is mean the resting cells. Endospore, as the name suggests, these are the intracellular resting cells. Okay, but how the endospores, these are different from the cyst formation? Here the cyst, here in the cyst we have discussed that the entire content of the bacterial cell it is utilized for the formation of the cyst. But the endospore, it is differing from the cyst in that only a part of the cell, only a part of the cell it is utilized in the formation, and not the whole cell. It is utilized for the formation of endospore. Okay, here in this one you can see it is the bacterial cell, and here the endospore formation takes place. Okay, these are the intracellular resting cells, and these are completely different from the cyst, because only a part of the cell it is utilized in the formation, and not the whole cell it is utilized in the formation of the endospore. Okay, the bacterial cell in which the endospore formation takes place. Okay, the bacterial cell in which the endospore formation takes place, these bacterial cells are referred as the sporangium. Okay, these bacterial cells in which the endospore formation takes place, these are referred as the sporangium. And the endospore formation, it is very common to the large sized bacteria. Okay, it is very common to the large sized bacteria. We have discussed that the bacillus or the rod shaped bacteria, these are larger in size whenever compared cocci and the spirilla. And that's why generally the endospore formation it is very common in the rod shaped or the bacillus bacteria. But the endospore formation it is also reported in very few of the cocci or the spherical and the spirillum bacteria, but very common to the bacillus or the rod shaped bacteria. Okay, typically here in this photograph you can see typically only one endospore is developed in one bacterial cell. Okay, typically one endospore it is developed in the one bacterial cell. Then their shape, their size, and the position it may vary in the different species. Okay, typically one endospore is developed in one bacterial cell, and their shape, size, and their position it may vary in the different species. For example, in this photograph here you can see the endospore. It is present in the center of the bacterial cell. Okay, here it is present at the terminal region of the bacterial cell, that is at one pole of the bacterial cell. And this one, it is in the third photograph, you can see it is subterminal. It is present near the one pole of the bacterial cell. Okay, that's why the endospore, regarding the position, it may be central, it may be terminal, or it may be subterminal. Okay, it may be central, terminal, or the subterminal. Okay, generally the endospore formation, it is mostly reported in the soil borne bacteria. Okay, soil borne bacteria. And these bacteria, generally these are pathogenic to the insects and the different types of the vertebrates. Okay, generally the endospore forming bacteria, these are soil borne bacteria, and these are pathogenic to the insects and the vertebrates. Okay, then the bacteria which are producing the endospores, these are secreting the certain type of the toxins which are causing the diseases. Okay, these are producing the toxins, and these are responsible for the certain type of the diseases. Okay, generally then the endospore formation, it is not takes place, it does not take during the active phase. Okay, during the active phase, that means in the first initial phases during the binary fission, generally they are multiplying very fast, the rate of multiplication is very fast. Okay, and generally the endospore formation, it never takes place in the initial active phases. Okay, in the initial active phases. And generally the formation of endospore, it takes place in the stationary phase. Okay, uh, at the stationary phase, that means the nutritional elements, that is, these are responsible for the formation of the endospore. That means whenever the nutrient availability is less, at that time the endospore formation takes place. Okay, as the endospore formation, it is occurred at the stationary phase. Generally it is believed that the endospore formation takes place due to the availability of the less amount of the nutrients. Okay, and generally the endospore formation, it is controlled by the protein metabolism of the cell. Okay, it is controlled by the protein metabolism of the cell. Okay, here you can see in this photograph in the center the structure of the endospore. Structure of the endospore, here the first of all in the center there is a presence of the core. Okay, this is the core, the central region of the central region of the endospore. This central core, it is having the large amount of protoplast, and it is mainly composed of DNA, then tRNA, ribosomes, and the different types of the enzymes. Okay, the center core, the endospore, in the center there is a presence of the center core, and this center core, it is made up of, it is having protoplast, and in this protoplast and the cytoplasm there is a presence of the DNA, then tRNA, and ribosomes, and the different types of the enzymes are present. Okay, and this center core, here you can see it is enclosed by the thin membrane, and this thin membrane, it is called as the core wall. Okay, the central core, it is enclosed by the thin membrane which is called as the core wall. Core wall, it is also referred as the germ cell membrane. Okay, it is referred as the core wall, and it is also called as the germ cell membrane. Okay, outer to this germ cell membrane, here you see there is a presence of the multi-layered structure, and that multi-layered structure is called as the cortex. Okay, outer side of the core wall there is a presence of the multi-layered structure which is called as the cortex. And the cortex, it is made up of the calcium dipicolinic peptidoglycan complex. Okay, the outer multi-layered cortex, it is made up of calcium dipicolinic peptidoglycan complex. Okay, then outer to the cortex, it is well protected by the presence of the spore coat. Okay, spore coat, it is a thick layer which is surrounding the cortex. Okay, it is surrounding the cortex. Generally the spore coat is differentiated into the outer layer and the inner coat. Okay, spore coat, it is differentiated into the outer coat and the inner coat. Okay, and this outer coat and the inner coat, it is made up of proteins. Okay, the spore coat, it is differentiated into the inner coat and the outer coat, and it is made up of proteins. Okay, it is made up of proteins. Then in the endospore, there is a presence of the additional covering layer is present, and this additional covering layer, it is called as the exosporium. Additional covering, or the outermost covering of the endospore, it is called as the exo means outermost. Here the outermost covering of the endospore, it is called as the exosporium. Okay, this exosporium, it is made up of polysaccharides, proteins, and the lipids. Okay, the exosporium, it is made up of polysaccharides, proteins, and the units proteins and the lipids. Okay, and these endospores, that is, these are very tough, these are resistance to the stress conditions. Okay, these are resistance to the stress conditions. Stress condition means whenever we can say the sufficient amount of moisture is not available for the bacterial cell, or whenever there is a very high temperature. These stress conditions, in such stress conditions, the bacterial cell can easily survive when the endospores are formed. Okay, and that's why, that is, these endospores, these can remain viable for the many years. Okay, these endospores, they can remain viable for the many years. Okay, the classic example of the endospore forming bacteria, that is the Bacillus anthracis. Okay, the Bacillus anthracis. Okay, Bacillus anthracis. Okay, the Bacillus anthracis, that is Bacillus anthracis in some of the country, that is, once it was used as a biological weapon. Okay, the endospores of the Bacillus anthracis, these are used as a biological weapons, because these are highly um, toxic. Okay, whenever the inhaling of such endospores of the Bacillus anthracis takes place, then it will readily attack the respiratory system of the human being, and ultimately in few minutes, even the death of the that person may takes place. Okay, that's why it is a Bacillus anthracis which is very toxic. That is the classic example of the endospore formation, and Bacillus anthracis, it may be used as the biological weapon. Okay, that is, in the past, the some of the American senator, they received the letter, and the letter, it was dusted with the endospores of the Bacillus anthracis. Okay, but it was then appropriate measures were taken, and they were sanitized, and they were then protected from the attack of the Bacillus anthracis. Okay, the Bacillus anthracis, the endospores, the inhaling of the endospore, it will readily attack the respiratory system of the human being, and even few minutes, the death of the that human, that person, it may takes place. Okay, that is why we are saying the endospore forming bacteria, these are highly pathogenic, and these are responsible for the certain type of the diseases, and even the death of the human beings. Okay, that is about the structure of the endospore. Okay, then here in the C, in this C photograph, you can see how exactly the germination of the bacteria takes place. Okay, in the endospore, it does not show the any detectable metabolic activity. Okay, these are not having the metabolic activity, but they remain viable for the many years. Okay, these are highly resistant to the heat, then ultraviolet radiations, then ionizing radiations, and the many toxic chemicals. Okay, these are highly resistant to the heat, ultraviolet radiation, ionizing radiations, and many toxic chemicals. And also these bacteria, these endospores forming bacteria, these are resistant to the physical and chemical agents. Okay, and generally their resistance, it is mainly due to the chemical composition of the cortex. Okay, the chemical composition of the cortex, that's why these are highly resistant to the stress condition. Okay, the chemical composition of the cortex, it is made up of calcium dipicylonic peptidoglycan complex. Okay, the germination, that is whenever the favorable conditions are available, whenever the favorable conditions are available, that means whenever these bacteria, whenever these endospores, these are getting the sufficient amount of nutrition, moisture, and the temperature, it will trigger the metabolic activities, metabolic reactions inside the endospore. It will trigger the metabolic reactions inside the endospores. Okay, the due to the imbibition of the water, or they will absorb the moisture, that is, they will swell, and ultimately their coat layers, the spore coat and the exosporium, it will get ruptured, and the new cell, it will grows out. Okay, new cell, it will grows out from the endospore. Okay, from this it is clear that from one endospore, only one bacterial cell is formed. Okay, from one endospore, only one bacterial cell is formed. And that's why some of the microbiologists believe that the endospore formation, it is not the process of multiplication, it is not the process of multiplication, but it is the process of perination. Process of perination means what? When the conditions are unfavorable, there is a formation of endospore, but when the conditions are favorable, then at that time one endospore will give rise to the one bacterial cell. That's why some of the scientists, some of the microbiologists believe that that is the process of perination and not the process of the reproduction. Okay,